Don't move, god damn it. Okay. Get ready for once a video because this video is going to contain everything that you need to know for blood in animal tissue in one shot. The topics that I'm going to cover right now already have their individual videos within this channel in English language indeed. However, this video is going to be a revision of all the chapters. So I'm not really going to write anything, just revise all the information that I have already mentioned in all those individual videos. So let's begin. Firstly, we are continuing with animal tissue. Under animal tissue, there are 4 major tissues, among which we are continuing with connective tissue. Under connective tissue, there's your connective tissue proper and specialized connective tissue. Under specialized connective tissue, there's your skeletal connective tissue and fluid or vascular connective tissue. Under fluid or vascular connective tissue, there's your blood and lymph. We are covering the blood portion of the animal tissue. So what is blood really? According to the chart that I just mentioned, blood is a specialized connective tissue. Why? Here's the thing. Connective tissue proper is proper because it contains all the typical features a connective tissue is expected to have. That is, connective tissue proper contains cells, fibers, and these cells and fibers, they are arranged on a ground called matrix. However, specialized connective tissue is special or different. How? For example, blood is a specialized connective tissue. Blood, out of the 3 major components of connective tissue, cells, fibers, and matrix, blood contains cells and these cells are present on a matrix. However, blood has no fibers, yet it is a connective tissue. Since this connective tissue is different, it's kept under specialized connective tissue. Not only that, blood has different names. First, blood is a fluid connective tissue. Why? Because the matrix on which the cells of blood lie is a liquid. Since the matrix, the ground on which the cells of blood, the blood cells are present, is fluid or liquid, it's called fluid connective tissue. Blood is also called vascular connective tissue because blood is found running inside tubes or vessels found inside our body, and these tubes or vessels inside of which blood runs are called blood vessels. There's a third name for blood as well. It's the circulating tissue in the body. Why? Because blood is responsible for circulation in the circulatory system of human body. The study of blood is known as hematology, where the heme part refers to blood and the logy part refers to study. And that's why the study of blood is known as hematology. We are going to study blood, or we are going through hematology under various topics one by one. So let's begin with the first topic, which is the formation of blood. The formation of blood is known as hemopoiesis, where the part hemo refers to blood and poiesis refers to formation. So the formation of blood is known as hemopoiesis. And the tissue that is responsible for hemopoiesis or the formation of blood is known as hemopoietic tissue. Now, the thing is that the hemopoietic tissue that is found inside human body differs with different stages of life. That is, the hemopoietic tissue before birth is different than the hemopoietic tissue after birth. So let's look at the different types of hemopoietic tissue present before birth. So before birth or during the time of gestation. Wait, what's gestation? Gestation is the time period between conception and birth. What's conception? When a sperm fertilizes an egg, that's called conception. And when baby comes out or the birth of the baby takes place, that's your delivery. So the time period between conception and delivery, that's your gestation. Or you could say it is the time spent by the developing baby inside the womb of the pregnant mother. Now, you might already know that the normal time period for gestation is 9 months. Let's divide this 9 months in 3 equal parts. Then we get the first 3 months, the middle 3 months, and the last 3 months. The first 3 months, the middle 3 months, and the last 3 months. In these different time intervals, the hemopoietic tissue found in the developing baby is different as well. In the first 3 months, the hemopoietic tissue found is the yolk sac. In the middle 3 months, it's your liver. And in the final 3 months, it's the spleen. After birth, right after birth, wait, let me manage the camera. Ooh. How about now? Yeah, I guess that's cool enough. So these 3 are present before birth or during the time of gestation. Right after birth, the hemopoietic tissue present is red bone marrow. Now, even during the time of gestation, among these 3 hemopoietic tissue, the major hemopoietic tissue is liver. Okay, now let's look at some of the basic features of blood. The total volume of blood found in human body, adult human body, is 5 to 6 liters. pH of blood is 7.35 to 7.45, so it's slightly basic. Arteries are the blood vessels that carry blood away from heart. So there's your heart, away from heart or towards your periphery. And veins are the blood vessels that carry blood towards heart. So from different parts of the body towards heart, the blood is carried by veins. Even blood, it's of 2 types. There's your oxygenated blood, which is rich in oxygen. So that's why it's bright red in color. And then there's your deoxygenated blood, which is scarce of oxygen. And that's why it's dark red in color. Some more basic features of blood are mentioned below. These will again be repeated with proper explanation under the topic of respiratory system. But let's try to understand it right now itself. 100 ml of blood contains 20 ml of oxygen at 100% saturation. What does 100% saturation mean? So your body contains blood. The blood contains red blood cells. The red blood cells contain hemoglobin. Hemoglobin is the respiratory pigment found in human blood. Hemoglobin is responsible to carry gases such as oxygen and carbon dioxide to different parts of the body. Now, the maximum capacity of one hemoglobin is to combine with 4 oxygen molecules. When one hemoglobin is combined with 4 oxygen molecules, that is when hemoglobin is at its maximum capacity with oxygen, then the blood is said to be 100% saturated because no more oxygen can hemoglobin handle or the blood take in. That's why it's 100% saturated. In that particular condition, 100 ml of blood contains in that particular condition, 100 ml of blood contains 20 ml of oxygen. Similarly, 100 ml of blood also contains 15 gram of hemoglobin. If you combine these 2 informations together, you come to find out 15 gram of hemoglobin contains 20 ml of oxygen at 100% saturation. That is when hemoglobin is combined with 4 oxygen molecules. In that particular condition, 15 gram of hemoglobin contains 20 ml of oxygen, because both 15 gram of hemoglobin and 20 ml of oxygen are present in 100 ml of blood when it's saturated 100%. So now let's move on to second topic. There's no stopping in the middle. We will just continue with what we have. Let me get rid of this. If you want to go pee, pause the video and go pee. If you want to go pee, don't pause the video and go pee. It doesn't matter. Okay, so now we know what blood is. Blood is a connective tissue. We also know how blood is formed. Blood is formed by the process of hemopoiesis in hemopoietic tissue. But do we actually know what blood really contains? We know what blood is, but what blood is actually? I mean, what is actually present inside blood? To know that, what you need to do is take a test tube filled with blood, put it in a centrifuging machine, and what does the centrifuging machine do? It spins rapidly. And as it spins rapidly, due to the centrifugal force, the heavier portion of the blood settles at the bottom of the test tube, while the lighter portion of the blood floats on top of the heavier portion. So, the end result of centrifugation would look something like this. So, there's your lighter portion on top of the heavier portion. You can see various layers in the test tube. There's your yellow top layer, which has a pale yellow color or straw yellow color. And at the bottom, there's your red part. In the middle of the yellow and red part, there's your very thin white line. Okay, so this pale yellow colored liquid is nothing but plasma. And plasma forms 55% of the liquid that is present in the test tube. Similarly, this red portion contains red blood cells, which forms 44% of the liquid present in the test tube. 100% minus 55% minus 44%. What's remaining? 1%. The remaining 1% is found sandwiched between plasma and red blood cells. This white layer contains white blood cells and platelets that make up 1% of the liquid present in this test tube. Sorry. So, out of 100% of what is present in the test tube, 55% is made up of plasma, and the remaining 44 plus 1, 45. 45% is made up of red blood cells, white blood cells, and platelets. Since red blood cells, white blood cells, and platelets, they are formed by the process of hemopoiesis, they are also called formed elements. So what's there in the test tube? In the test tube, there's 55% straw yellow colored plasma and remaining 45% formed elements. Similarly, with this, we can also conclude that in human blood as well, there's 55% plasma and remaining 45% formed elements, also called blood cells. Okay, so now we know what the different layers are. What we are going to do now is study all these individual layers one by one, starting off with plasma. We now know that plasma is a pale yellow colored liquid that makes a majority portion of the blood, more than 50%. But what is plasma actually composed of? Majority, and by that I mean 92% of what's present in plasma is just plain old H2O. That is why blood is known as fluid connective tissue, because majority portion of the blood is just water, and the cells present in the blood are present on this watery layer or liquidy layer. That's why it's called fluid connective tissue. 100% minus 92% gives 8. So the remaining 8% contains other substances, and these other substances are either inorganic substances or organic substances. Under inorganic substances, you can find salts of sodium, potassium, such as. So inside your blood vessel, there's your blood. Majority portion of blood is just plasma. In that plasma, you can find organic and inorganic substances. First, we are looking at the inorganic substances. So the inorganic substances found are, first, the salts. Salts of sodium, potassium, like sodium chloride, potassium chloride, sodium bicarbonate, potassium bicarbonate. All these salts have their significance, and you'll come to know their significance under the chapter of respiratory system under transportation of gases. There are also gases, because blood is the one transporting them. like oxygen, carbon dioxide, nitrogen. In bad cases, very bad cases, carbon monoxide. That's your inorganic substances. The organic substances present are number one, plasma proteins. The proteins that are found in the plasma of the blood are called plasma proteins. There are various plasma protein, but the three major plasma proteins that you need to know are albumin, which is responsible for osmoregulation, clotting factor, which is responsible for blood clotting or healing of wound. There are two major clotting factors, fibrinogen and prothrombin, found in your blood. Then immunoglobulins. These are globulin proteins that make up the immune system of the body, so-called immunoglobulins. And they are nothing but the antibodies found inside our body. Then there are monomers. What are monomers? The food we eat contains polymers, such as protein, fats, carbohydrates. These are large and can't get absorbed by our body. So these are first broken down chemically and mechanically by our digestive system and finally converted into absorbable form called monomers, such as for protein it's amino acid, for carbohydrate it's glucose, for fats it's fatty acid and glycerol. They, once absorbed by the digestive system, enter into the blood and through blood get assimilated or distributed to different parts of the body. Waste product. Nitrogenous waste products like urea, uric acid, hormones. The endocrine glands found inside our body secrete hormones, and these hormones are secreted directly into the bloodstream, and in the bloodstream they travel through this plasma and reach their target organ. Thyroxine, produced by thyroid gland, progesterone, all these hormones, they are secreted directly into your blood. And finally, enzymes. All this stuff that I have mentioned here are found in plasma and is what plasma is composed of. So we are done with the first layer of what's present in the test tube. Now let's begin with the second layer, or the red blood cells. Okay, so the paper is fixed and we are ready to go. I have no idea why I'm coughing so much. I'm sorry if you are disgusted by it, but that's not my problem. Let's go. Okay. So the second person, which makes 44% of what's present in your blood, is red blood cell. A red blood cell, or RBC, is also called erythrocyte, where the erythro portion means red and the cyte portion means cell. So the cells that are red blood cells. There you go. Okay, so we are gonna study the various formed elements, by that I mean red blood cells, white blood cells and platelets, under various titles to make it easy. And at the same time, I want you to compare the information under each title of all these three formed elements. So firstly, erythrocyte. The first title is formation of erythrocyte. Inside our body, in blood, there's your erythrocytes. Erythrocytes are there because it's formed from somewhere. And the process by which these erythrocytes are formed is called erythropoiesis, where erythro part refers to red blood cell and poiesis part refers to formation. So the formation of red blood cells are called erythropoiesis. Let's look at it. Firstly, our body contains kidney. Inside kidney, there are these special cells that produce a hormone called erythropoietin, where erythro refers to red blood cell and poietin refers to the hormone that brings about poiesis or formation. So the hormone secreted by the special cells found inside your kidney that is responsible for erythropoiesis, or that is responsible for the formation of red blood cells, is erythropoietin. This erythropoietin goes to your long bone. Inside your long bone, there's a long shaft in the middle called diaphysis and two knob-like structures at the two ends called epiphysis. Inside epiphysis, you can find red bone marrow. Erythropoietin kicks the red bone marrow and tells it to start the process of erythropoiesis. So erythropoiesis after birth takes place in red bone marrow. And for erythropoiesis to start in red bone marrow, who is responsible to kick it off or provide a starting kick? It's the hormone called erythropoietin, which is secreted by the special cells found inside your kidney. Okay, so once the red bone marrow is kicked, it starts the process of erythropoiesis. During erythropoiesis, inside red bone marrow, check the camera. Okay, that's fine. Inside red bone marrow, there are these large cells with all the cells organelles, including nucleus, called erythroblast. Erythro refers to erythrocyte or red blood cell, and blast means precursor cell. Precursor cell of, suppose, cell A is that cell which is responsible for the formation of cell A. What do I mean? Okay, let's suppose there's cell A for animal. Cell A. And then there's this cell which is the precursor cell of cell A. It means that for the formation of this cell A, this precursor cell of cell A is responsible. From this precursor cell, the cell A has been formed. Similarly, from this erythroblast, erythrocyte is formed. That's why erythroblast has been given the name erythroblast, where blast literally means precursor cell, the cell that gives rise to other cells. Whew. Okay, erythroblast divides and multiplies and finally form this cell, which contains all the cell organelles as that of erythroblast, but along with the cell organelles, the major component of red blood cell has started to form. So you can find that major component in minute traces. And what is that major component? It's your hemoglobin. So in fine traces, you can find hemoglobin as well inside this cell. And this cell right here is known as reticulocyte. Reticulo, it's like network. Reticulate means there's something inside that's tangible, that's kurkura, and that's your cell organelle. Cyte means cell. Reticulocyte is your immature RBC. Once reticulocyte matures, the formation of mature RBC takes place, and mature RBC is known as erythrocyte or red blood cell. But for reticulocyte to mature into an erythrocyte, there needs to be stuff inside your body obligatorily. What are those stuff? So inside your body, there must be folic acid, vitamin B12 or cyanocobalamin, and iron. These three components are necessary for reticulocyte to mature into erythrocyte. If any one of these three components is lacking, then erythrocyte won't be formed from reticulocyte. Along with these three stuff, there also needs to be Cassell's intrinsic factor. What's Cassell's intrinsic factor? Okay, suppose you take a vitamin B12 supplement, eat it. Once that vitamin B12 supplement reaches your stomach produces something called Cassell's intrinsic factor, which you'll come up to know again in the chapter of digestive system. But for now, your stomach produces Cassell's intrinsic factor. Cassell's intrinsic factor is responsible for the absorption of vitamin B12. That is, once the vitamin B12 reaches your stomach, it gets absorbed with the help of Cassell's intrinsic factor. Let's suppose your stomach stops producing Cassell's intrinsic factor. Then you take in a vitamin B12 pill. It's of no use now, because vitamin B12, after reaching your stomach, won't be absorbed by your body. That means the vitamin B12 comes out in the form of poop. So vitamin B12 is basically useless without Cassell's intrinsic factor. So after all these requirements are met, reticulocyte converts into erythrocyte. Once reticulocyte converts into erythrocyte, changes take place. Like change number 1, the size of the cell becomes very small. Change number 2, all the cell organelles that were present in reticulocyte, they disappear, including nucleus, and this I'm talking about inside human body. Including nucleus, all the cell organelles, they disappear. So no cell organelles are found inside erythrocyte. There's no nucleus, no endoplasmic reticulum, no mitochondria, none of that. And erythrocyte is nothing but a bag filled with hemoglobin. So there's your Donans membrane, which is the outer membrane, and inside of which there's just hemoglobin. It's just filled with hemoglobin, which is the respiratory pigment. Okay. So now we know how red blood cell is formed. Now let's look at the shape of the red blood cell that has just formed. If we look at this red blood cell from front, then it looks round. And if you cut it in the middle, cut it from the middle and look at it from the side, then it looks biconcave. That is, there is concavity from both sides. And that's because in the middle, there's no nucleus. Due to the absence of nucleus, both the sides have depressed inside, or there's biconcavity. And this particular red blood cell is what's found in mammals. I repeat, in mammals. Inside mammals, the mature mammalian RBC is round, biconcave, and enucleated or without nucleus. However, inside all other vertebrates except mammals, all other vertebrates, including exception, that is camels and llamas. Camels and llamas, they are mammals, yet they have exceptional RBC. That is, the RBC inside camels and llamas are the same as that of all other vertebrates except mammals, including frogs. They contain oval, not round, oval, biconvex, not biconcave. It is swollen from both sides, so it's biconvex. And nucleated, it's biconvex or swollen from both sides because there's a nucleus in the middle. That's why. So mature mammalian RBC is round, biconcave, and enucleated. Mature RBC in all other vertebrates except mammals and including exceptional mammals like camels and llamas, the RBC found is oval, biconvex, and nucleated. Okay, so what's the advantage of biconcavity of RBC? Here's the advantage. Let's suppose you have two RBCs or red blood cells and both of them have same volume, but one is biconcave and the other is biconvex. The RBC that is biconcave has a higher surface area compared to the RBC that is biconvex having the same volume. So for the same volume, surface area is high. That is, surface area to volume ratio is high. That is, the respiratory efficiency is high. That is, on the surface there are more and more hemoglobin. That is, more and more oxygen can get combined with hemoglobin. And that is why respiratory efficiency is high. Next is RBC count. The normal number of RBC, I repeat, normal. The normal number of RBC in males is 5 to 5.5 million per cubic millimeter of blood. 1 cubic centimeter refers to 1 milliliter, so you can calculate how much 1 cubic millimeter means. So 1 cubic millimeter of blood contains 5 to 5.5 million red blood cells in males. In female, however, it's 4 to 4.5 million red blood cells per cubic millimeter of blood. The question arises, why is the number of red blood cells found in female much less than the number of red blood cells found in males? There are two major reasons. Reason number one, inside female, there's no testosterone. What's the significance of testosterone? Inside male, there's testosterone, and testosterone is also responsible for the process of erythropoiesis. That is, higher the testosterone, more erythropoiesis and more red blood cells. Since females, they don't have testosterone, erythropoiesis takes place, but at a lesser amount as compared to males. That is why females have red blood cells at a lesser number than males. And number two, female goes through menstrual cycle every month where there is a heavy loss of blood, and due to menstruation, the number of RBC found inside female is much less than the number of RBC found inside males. Second, if the number of RBC is more than or less than the normal number, then that's your abnormal condition. If the number of RBC is less than the normal number, it's called erythropenia or anemia, where penia, you could use it to remember that penia means number is less. If you see penia, it means the number is less than the normal number or the borderline number. If the number is more than the normal number, it's your polycythemia. Let me give you an example of polycythemia. People living at high altitudes, let's say in Himalayas, people living in Himalayas, they have red cheeks. Why? Because at a higher altitude, the atmosphere is thin. That is, the percentage of oxygen in atmosphere is very low. So the people living there have to survive in an atmosphere with low percentage of oxygen. And as an adaptational feature, people living there have higher number of red blood cells due to higher erythropoiesis, and due to which they have higher amount of hemoglobin. Since they have higher amount of hemoglobin, they can take in more and more oxygen from that small percentage of oxygen that is present in atmosphere. And since they have higher amount of hemoglobin, which is responsible for the redness of the blood, their cheeks, they look red. And this particular condition is polycythemia. Average lifespan. After 120 days, red blood cells have become old enough to get destroyed. And they get destroyed by the process of hemolysis, where hemo refers to blood and lysis refers to breakdown. So after 120 days, the red blood cells first get phagocytosed or eaten up by white blood cell, and then the remaining portion gets destroyed by spleen, liver, red bone marrow, etc. Out of all these three, the place where maximum number of red blood cells get destroyed every second is spleen, and that is why spleen is also called graveyard of RBC. But spleen is also called blood bank. Now, looking at it from surface, it seems that these two names are completely different, because one's talking about killing and another's talking about producing. But here's the thing. Spleen is called graveyard of RBC because the maximum number of old red blood cells get destroyed per second in spleen. However, spleen is called blood bank because the new RBCs that are made by the process of erythropoiesis get stored in the spleen. That's why it's a bank of blood bank, due to the property that it stores new blood cells or red blood cells. The respiratory pigment found inside a red blood cell is hemoglobin. It's called a respiratory pigment because hemoglobin transports gases that are required for respiration. Hemoglobin contains two parts. There's your heme part, and then there's your globulin protein part. Looking at the structure of hemoglobin, it looks something like this. So there are 4 iron. These 4 iron, they make up the heme part, and heme part is what imparts a red color to blood. Surrounding each iron, there's your protein called globulin protein. Out of the 4 globulin protein, 2 are beta globulin protein and other 2 are alpha globulin protein. During the transportation of gases, the gases such as oxygen and carbon dioxide, they get attached to the iron part or the heme part. Function. Since red blood cell contain respiratory pigment, the function of red blood cell is to transport oxygen and carbon dioxide for respiration. So we are done with the first formed elements. That's your red blood cell. Yite, 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 yite. Then there's your second. There's your second. I'm gonna go drink water and come back. See you. Whew. I am back, baby. So the second formed element is white blood cell, which is also called leukocyte, where leuko means white and cyte means cell. So the cells that are white blood cells or leukocyte. Let's look at leukocyte the same way we studied erythrocyte. First, the process by which leukocytes are formed is called leukopoiesis, where leuko refers to leukocyte or white blood cell and poiesis refers to formation. So the formation of leukocyte or white blood cell is leukopoiesis. And leukopoiesis takes place in the same place, at the same place where erythropoiesis took place, that is red bone marrow. Along with that, lymph nodes also form white blood cells. Second, shape. In case of red blood cells, the shape of the red blood cells were definite. That is, they were round, biconcave, and without nucleus. In case of human. However, the white blood cells are of variety of shapes. Some white blood cells, they look like amoeba. That's why they are amoeboid in shape. Some are round, so rounded. Some don't have definite shapes, so they are irregular. Thirdly, the number of red blood cells, they were 5 to 5.5 million per cubic millimeter in the blood of male and 4 to 4.5 million per cubic millimeter in the blood of female. WBC count, on the other hand, is 5,000 to 10,000 per cubic millimeter. That is, in 1 cubic millimeter of blood, you can find only 5,000 to 10,000 white blood cells, only in comparison to red blood cell, because there's a red blood cell that's found in millions per cubic millimeter, and then there's your white blood cells that's found in thousands per cubic millimeter. So very low as compared to red blood cells. Abnormal condition is when the number is more or less than the normal number. If the number of white blood cells is less than the normal number, it's called leuko. Leuko refers to white blood cell. And what did I say you to remember? When the number is less, it's penia. So leukopenia. If the number of white blood cell is more than the normal number. Now, there are two reasons why this could occur. That is why the number of white blood cells could be more than the normal number. One is during infection. For example, there's a bacteria and this bacteria enters your blood. To destroy this bacteria, your body prepares or makes more white blood cells and send them to destroy that bacteria. So your body is infected, and to fight against that infection, your body makes more white blood cells. Maybe during dengue as well, if you got tested for dengue, you might have done the CRP test. I think that's what it's called. That's also related to infection. Anyway, I don't know much about it. During infection, the white blood cell count inside human body increases. And during that condition, it's called leukocytosis. Remember, cytosis refers to number being more than normal. But if the white blood cells found inside the blood start dividing uncontrollably, that is, they start dividing rapidly, then you get cancer. Since the cancer is present in the blood, it's called blood cancer. And since this blood cancer is due to leukocyte, it's called leukemia. Or blood cancer is leukemia. Then average lifespan, 8 to 10 days. Diapedesis. So diapedesis is a special feature that is present in white blood cells. Suppose here's your capillary, right? Here's the thin wall of the capillary. Outside capillary, here's your bacteria. Now, somehow your body detects this bacteria and sends white blood cell to kill or engulf or phagocytose or eat up this bacteria. These white blood cells that are present inside capillary squeeze out through the thin wall of the capillary, and this property or this process by which white blood cells squeeze out of the thin wall of the capillary during infection or also during the formation of lymph is known as diaperidesis. Then pigment. White blood cells, they have no pigments. That is, there's no hemoglobin. Function. White blood cells are the soldiers found in human body. They are responsible for the immune system of your body. Nucleus. RBCs, they didn't have any nucleus. However, white blood cells, they don't just have nucleus, they have nucleus of different types. Now let's look at the different types of white blood cells. I think that's good enough. Okay. Types of white blood cells. White blood cells are of two types depending on their granules or not. That is, on the basis of presence or absence of granules inside the white blood cells, they are of two types. There's your granulocyte. Granulo refers to granule and cyte refers to cell. So granulocytes are those white blood cells that contain granules inside of them. And then there's your agranulocyte, where A refers to absence. So agranulocytes are those white blood cells that don't contain any granules. Granulocyte have a special name of their own, and it's polymorphonuclear leukocyte. I know it's a long name. Don't get intimidated by it because I'm going to make it easy for you. Polymorphonuclear leukocyte. Let's break this large name into pieces. So there's your poly, then morpho, then nuclear, and then leukocyte. Poly means multiple. Morpho means morphology or form. Nuclear means nucleus. And leukocyte means white blood cell. So granulocytes are those leukocytes or white blood cells that contain nucleus of poly or multiple forms or morphology. That's why granulocyte is called polymorphonuclear leukocyte, because granulocytes are leukocytes that contain nucleus of multiple morphology or multiple forms. Okay, granulocyte, on the basis of the type of granules found inside cells, there are of three types. 1, 2, 3. In the first cell, you can see that the nucleus that is present has multiple lobes. In this particular diagram that I have drawn, the nucleus has 1, 2, 3, 3 different lobes, and these lobes are connected with the help of connective handle. And this granulocyte that contains multi-lobed nucleus is called neutrophil. Why? Neutro means neutral and phil means love. So basically what it means is that this particular cell loves both acidic and basic dye equally, or the love of this cell right here is neutral towards both acidic and basic dye. For it to be neutral towards both acidic and basic dyes, it must contain both acidic and basic granules. What are acidic and basic granules? Acidic granules are those granules that turn red once acidic dye is put on it. Basic granules are those granules that turn blue once basic dye is put on it. Since neutrophil contains both acidic dye and basic dye, it gets colored or stained by both acidic, since neutrophil contains both acidic granules and basic granules, the cell gets stained by both acidic and basic dyes. Since neutrophil gets stained by both acidic and basic dyes, neutrophil loves both acidic and basic dyes equally or neutrally, and that's why it's called neutrophil. Let's suppose there are 100 granules inside neutrophil. Then out of 100 granules, there's a possibility that 50 of them are acidic granules and the 50 of them are basic granules. So if you put acidic dye on it, then the 50 acidic granules get stained red. But if you put basic dye on it, then the 50 basic granules get stained blue. And since only the 50% gets stained when one of the two dyes are put on it, the staining is weak. Okay, the function of neutrophil is phagocytosis. Whenever a bacteria enters your body or a pathogen enters your body, the first soldiers that are sent to fight against these pathogens are neutrophils. The number of neutrophil increases than normal number. Here's your normal number. The number of neutrophil becomes more than the normal number if you exercise. During exercise, that's why exercise is important for your health. You can see a concrete example or concrete evidence right here itself. When you exercise, the number of soldiers fighting pathogens inside your body, that is neutrophils, increases and your immune system boosts up or becomes better. Secondly, during pregnancy, a woman has to face a lot of mental and physical stress during such stressful time. To make sure that the body of the woman is able to fight against pathogens, the number of neutrophil inside the body of the woman increases. And that particular condition where the number of neutrophil is more than the normal number is called neutrophilia. So there's phil or love towards neutrophil. So neutrophilia. Secondly, there's your white blood cell, which contains bilobed nucleus. That is, the nucleus contains two lobes, and these two lobes are connected with the help of a connective handle. And upon viewing the nucleus, it looks like a spectacle or glasses. And this particular cell with bilobed nucleus is acidophil or eosinophil. Let's break the word. Acidophil. Acido refers to acid and phil refers to love. It's called so because this white blood cell loves acidic dye. That is, when you put acidic dye on this cell, it gets stained red strongly. The reason is the granules found in this cell is acidic. That's why they get stained red once acidic dye is put on it. And the acidic dye that is used to stain this cell is called eosin. That's why it's also called eosinophil, where eosino refers to eosin and phil means love. That is, this particular cell loves eosin or gets stained strongly with eosin. Function. Acidophil or eosinophil mediates inflammation. During inflammation, who is responsible? Histamine is responsible. And mediating inflammation is done by acidophil or eosinophil. Whenever an allergen is something that causes allergy. Whenever an allergen is introduced to your body or enters your body, you get allergy. At the point where allergy has occurred, inflammation occurs. And at that inflammation, the thing that is responsible to mediate, not increase, not decrease, mediate the process of inflammation is acidophil or eosinophil. And that particular condition where the number of acidophil or eosinophil is more than the normal number, such as during allergy, during asthma, during infection, that particular condition is known as eosinophilia. That particular condition is eosinophilia. Third. So the third granulocyte contains nucleus that is shaped like the alphabet or the letter S. So the granulocyte with S-shaped nucleus is called basophil, where baso means base and phil means love. That is, this cell loves base. That is, whenever basic dye is used to stain basophil, it stains strongly because the basic granules present in the basophil, they get stained blue when basic dye is put on it. The function of basophil is similar to that of mast cells found in connective tissue, and that is, it is secretory in function. What do they secrete? They secrete histamine that causes inflammation, serotonin that decreases inflammation as well as it's a neurotransmitter, and heparin, which is an anticoagulant or prevents blood clotting. The condition where the number of basophil is more than the normal number is called basophilia. So that's it for granulocyte. Let's move on to agranulocyte. Under agranulocyte, there are two cells. There's your monocyte, where mono refers to single and cyte refers to cell. So it's a single cell. Monocyte, if you look at the diagram and compare this diagram with the rest of the cells, you can see that the monocyte is very large. And indeed, it is the largest WBC found in human body. Monocyte is the largest WBC found in human body. The type of nucleus that is present inside this monocyte is kidney or bean shaped. That is, the nucleus is shaped like a kidney or a bean. The function of monocyte. Monocyte, they fuse and form macrophages. Macrophages, macro means large, phase means the cell that shows phagocytosis. So these are large phagocytic cells. They are responsible for eating up pathogens. If neutrophil, that are the soldiers sent first for the process of phagocytosis, can't do their job, then macrophages are sent, which are much bigger and much stronger, so they can easily defeat pathogens. The macrophages found in different parts of the body have their own names. For example, macrophages found in alveoli are called dust cells. Macrophages found in liver are called Kupffer cells. Macrophages found in central nervous system, such as your brain, are called microglia. And macrophages found in bone are osteoclast. Macrophages found in connective tissue are histiocytes. When the number of monocyte is more than the normal number, then that's called monocytosis. Second is lymphocyte. Lympho refers to lymph and cyte refers to cell. So they are called lymphocytes because lymph is composed of lymphocyte, plus the lymph nodes, they produce lymphocytes as well. If you compare the diagram of lymphocyte with the rest of the cells, you can see that lymphocyte is the smallest one present here. And indeed, lymphocyte is the smallest white blood cell present in human body. The nucleus that is found is spherical, and as compared to the size of the cell, the size of the nucleus is large. So the amount of cytoplasm is very less or scanty. There are two types of lymphocytes. So here's your long bone. Inside the long bone, there's your bone marrow. Inside bone marrow, the process of formation of white blood cells take place. During the process of formation of white blood cells, lymphocytes also form. If the lymphocytes, they mature within the bone marrow, then they come out to be B lymphocytes. And B lymphocytes, they produce antibodies such as immunoglobulin A, immunoglobulin G, immunoglobulin M, immunoglobulin E, immunoglobulin D. These are the various antibodies that are found inside human body. These antibodies, they are made and sent by your B lymphocytes. Since these antibodies are present in the humor or the fluid portion, such as blood and lymph of human body, and since these antibodies, they provide immunity, the immunity gained from B lymphocytes or the antibodies produced by B lymphocyte is called humoral immunity. But if the lymphocyte comes out due to the secretion of thymosin by the endocrine gland called thymus. Here's the thing. So there's an endocrine gland named thymus. This endocrine gland produces a hormone called thymosin. This thymosin makes some of the lymphocytes from the bone marrow to come out. And these lymphocytes, they enter the thymus get trained there. That's why thymus is called training school of T cells. They get trained there and come out in the form of various types of T lymphocytes like helper T cells, killer T cells, etc. So the lymphocytes that came out of the bone marrow and matured inside thymus, these lymphocytes are T lymphocytes. These T lymphocytes of various types, such as helper T cells, killer T cells, they are cells, and these cells are responsible for providing immunity. Since cells are providing immunity, that particular immunity is called cell-mediated immunity. And the condition where the number of lymphocyte is more than the normal number is known as lymphocytosis. If you find this lecture to be too fast or high-paced, I recommend you watch individual videos under each of these titles, because this is a revision video, not a concept building video. Oh, where's my chappal? Nah. Oh, here it is. Time for the last formed element. I'm gonna go drink some water and come back. And I am back. Let's go. Let's go. Let's complete this now. So the last formed element that we have remaining to do is thrombocyte and platelets. Now you might have So you might have figured out that I just said thrombocyte and platelets and not thrombocyte or platelets, because thrombocyte and platelets are actually not the same. There's a difference between these two. And what's the difference? Let's learn about it. First, platelets. The process by which platelets are formed is called thrombopoiesis, where thrombo refers to either platelets or thrombocyte and poiesis refers to formation. So the formation of thrombocyte or platelets is thrombopoiesis. But thrombopoiesis of platelets is different. Thrombopoiesis of platelets, this is what takes place. Firstly, inside the red bone marrow, there's the long bone. This is the God damn it, I didn't tape my paper. That's why it's moving so much. Okay, so here's the epiphysis region. Inside the epiphysis of the long bone, there's your red bone marrow. Inside the red bone marrow, there are these large cells with large nucleus called megakaryocyte. What are they called? They are called megakaryocyte. Mega refers to huge, large, karyo refers to nucleus, and cyte refers to cell. So these are cells with large nucleus, so-called megakaryocyte. The megakaryocyte, they enter blood, or shall I say, try to enter the blood. However, the megakaryocyte is too large to penetrate through the wall of the blood vessel. So as this megakaryocyte tries to penetrate through the wall of the blood vessel, the cytoplasm of the megakaryocyte breaks down into many smaller fragments. And these small fragments of cytoplasm of megakaryocyte are called platelets. So platelets are actually not cells. They are the cytoplasmic fragments of megakaryocyte. And as these fragments are formed, the nucleus gets destroyed. That is why platelets are the cytoplasmic fragments which are enucleated or without nucleus. Shape. Platelets are very small because it has been formed by the breakdown of a cell of the cytoplasm of a large cell. They are irregular. They don't have definite shape. They are irregular, enucleated without nucleus, sticky, chip-chipa, that is they stick easily, and fragile, that is they break easily. Number. It's 200,000 to 400,000 per cubic millimeter. More than white blood cells, but less than red blood cells. Average lifespan 7 to 10 days. Abnormal condition. When the number of platelets is less than the normal number, then that particular condition is called thrombocyto, where thrombocyto refers to platelets. And what did I tell you to remember for something for a number that is less than normal count? Penia. Erythropenia, when the number of RBC is less than normal. Leukopenia, when the number of WBC is less than normal. And thrombocytopenia, when the number of platelets is less than normal. When the number of platelets is more than the normal number, then that's your what did I tell you to remember? Cytosis. When the number of white blood cells is more than the normal number. Leukocytosis. When monocytosis. When lymphocytosis. Similarly, when the number of platelets is more than the normal number, that's your thrombocytosis. Pigment, there are no pigments found in platelets. Function. The major function of platelets is blood clotting or healing of wound. Suppose you just got scratched, and from the area where you just got scratched, blood starts to come out. After some time, the area gets healed, and that's due to platelets. If there were no platelets, the clotting of the blood would not have occurred, and your wound would not have healed, and you would have bled to death. Second is serotonin. And serotonin is anti-inflammatory because it is a vasoconstrictor. Vaso refers to blood vessel and constrictor refers to constriction or thitab. So the blood vessel gets constricted and the swelling decreases. Okay, so that's your platelets. What's different about thrombocyte from platelets? Here's the difference. Platelets are the clotting fragments found in mammals. So in mammals, what you have are called platelets. But in all other vertebrates except mammals, you have thrombocyte, where thrombo refers to clot and cyte refers to cell. So the clotting cells found in all vertebrates except mammals, because in mammals there are platelets, except mammals are thrombocytes. Platelets don't have regular shape, they are irregular, and they don't have nucleus, they are enucleated. However, thrombocytes are actually cells and they have fixed shape, as you can see here. The shape of thrombocyte is spindle shape, that is, it is swollen in the middle and tapering at the ends. Spindle cell, and it is swollen in the middle due to the presence of nucleus in the middle. So here's your nucleus in the middle of the thrombocyte and it's tapering at the end. So the shape is spindle shaped and it is nucleated. So now we know what platelets are. And we also know that platelets are responsible for wound healing or blood clotting inside human body. But how does blood clotting take place, or how does a wound heal? The thing is that the process of blood clotting is actually a really vast and complicated process. As many as 14 factors and vitamin K are involved for this process. However, what we are going to see here is the oversimplified version of what actually takes place inside the human body. This is enough for your entrance level examinations. So let's continue. Blood clotting. Here's your blood vessel. Suppose you get pricked or injected with a needle at this point on your blood vessel. As a result, your blood vessel suffers a puncture. From the area or the point, which is the site of injury, where the blood vessel has been punctured, blood starts to come out or bleeding takes place. This area needs to be healed so that the person doesn't die of blood loss. And the process by which this area gets held is called blood clotting. Clotting means coming together or getting aggregated at one particular place. And since the aggregation takes place of the blood cells, it's called blood clotting. Blood clotting takes place in two steps. First is temporary clotting, where the bleeding gets stopped temporarily. And second is permanent clotting, where the bleeding gets stopped permanently. Immediately, temporary clotting takes place. Where? This is what happens. So here's your site of injury from where the blood is coming out or bleeding is taking place. Inside your blood vessel, there are platelets. These platelets immediately come at the site of injury and stick with one another or fit with one another. Since they are small, a large number of platelets can come aggregate here. Since they are irregular in shape, they can easily fix on one another. And since they are sticky, they can stay together or stick together easily. As a result, they cover this area up. And this area is covered due to the cohesion of platelets. Once this has happened, the bleeding stops temporarily. After temporary clotting, the process of permanent clotting starts. So, inside your blood, there's something called anticoagulant. And anticoagulant doesn't like clotting. Anticoagulant is anti or against coagulation or clotting. When there's already someone that is against clotting inside your blood, how does clotting even take place? Well, it takes place because that particular someone, that is heparin, heparin is the anticoagulant that is present inside your blood, gets anticoagulant. This heparin, at the site of injury, comes in contact with air, and the oxygen of the air oxidizes the heparin. As a result, heparin becomes inactive, and the process of clotting can continue. So one problem is sought out. Second, the platelets that have aggregated at the site of injury rupture because they are fragile, and as they rupture, they secrete something called thromboplastin. This thromboplastin, with the help of calcium present inside the blood, gets converted to thrombokinase. And thrombokinase is very essential because thrombokinase is what activates the inactive clotting factors or clotting proteins present inside the blood. Since thrombokinase is important, the stuff that converts thromboplastin to thrombokinase is important, and that particular stuff is calcium. So calcium is not just important for bones or muscle contraction, it's also responsible for blood clotting. Thrombokinase then activates the first clotting protein called prothrombin. Prothrombin is inactive. Thrombokinase activates it to thrombin. You can figure out that prothrombin is inactive with the help of this part, pro. Prorenin is the inactive form of renin, the same way prothrombin is the inactive form of thrombin. This thrombin then activates the second clotting protein, which is fibrinogen, present in your body. It is inactive, you can figure out with the help of gen. Trypsinogen is the inactive form of trypsin, the same way fibrinogen is the inactive form of fibrin. Fibrinogen initially is present in the form of individual fibers like this, individual fibers in the blood. Once fibrinogen gets activated with the help of thrombin, it forms fibrin. And fibrin is actually a network of the fibers. So here's your fibers that are present individually. These fibers, they form network. And this network or maze of fibers is known as fibrin. It's a network. At the spaces between inside or in this network, these spaces, they get filled with dead cells. So the dead cells, they go and stay here and form a handiplast-like structure or clot or thrombus. This clot and or thrombus covers this place up or patches this place up, and permanent clotting takes place and the bleeding permanently stops. After that, the layers on top of this permanent clot that have injured get closed with the help of fibers, which is secreted by the fibroblast cells. And finally, the injury gets plastered. Now, prothrombin is an essential clotting protein present inside your blood. And this prothrombin is secreted by your liver. For liver to make and secrete prothrombin, it needs vitamin K. And that's why vitamin K is very, very essential for blood clotting. If there was no vitamin K, if there is no vitamin K inside your body, liver won't be able to produce prothrombin and the process of blood clotting won't take place. In fact, in absence of any one of the 14 factors, or in absence of vitamin K, your body is unable to complete the process of blood clotting, and the area of injury doesn't get clot or doesn't get covered up. As a result, bleeding continues and continues and continues, and the person loses a lot of blood, and this can even lead to death. So this particular condition where the wound just doesn't agree to get healed up due to the absence of either one of the 14 factors or vitamin K is called bleeding disorder, called hemophilia. It is a condition of non-stop bleeding. That is, the person with hemophilia, once he or she gets injured, the person keeps on bleeding. The bleeding just doesn't want to stop. And this takes place when any one of the essential components required for blood clotting is missing. And that's it for blood under the chapter of animal tissue in one shot. Next is going to be a one shot video for blood groups, under which I'm going to show you teach you blood ABO blood grouping, RS blood grouping, RS incompatibility, and overall what antigens and antibodies even mean to begin with. See you again.